So it's my pleasure to introduce the afternoon's first speaker, Botond Roska. He earned his PhD in molecular and cell biology from UC Berkeley, and he's presently a professor in the Faculty of Medicine at the University of Basel in Switzerland, and he's also a senior group leader at the Friedrich Meister Institute for Biomedical Research, also in Basel. And he is a member of the Inscopics SHB. And his laboratory has been using interdisciplinary approaches combining molecular biology, physiology, virology, and computational neuroscience to look at the structure and function of neural circuits in health and disease, particularly in the visual system, retina, and also other visual areas. Very pleased to have you today, Botond.
Thank you very much for the invitation. It's a really pleasure to be here. It's wonderful to see Inscopics growing and being at this stage. So vision starts with a movie that enters our eye and captured by the retina. And from this, it flows to a number of brain regions. Probably for us, for perception, LGN is most important. From here to the primary visual cortex, and then to 30 or 40 different visual areas. And despite this very large complexity of brain areas and cortical areas leading to a percept, indeed, most diseases, 95% or even more of visual diseases, are localized at the very beginning, in your eye, in the retina. So if we look into the eye, what we see is an amazing machine, what we call a retina. This is a parallel image processor. What it does, it has one input, a video that enters the retina. And in all mammals and in many vertebrates, what you have is around 20 or 30 movies that coming out from your eye and pouring towards the LGN and other visual centers. Now, if you look in this device, and Ramon y Cajal already did this a number of years ago, what he found was a zoo of different morphologies. And not only you see different cells, but they're coming repeatedly, and this was the idea of a cell type. And there are approximately 70 or 80 such morphologies in the retina, and they organize into circuit mosaics. So in this circuit mosaic, you have a few elements clustered at the different points, and this is acting as basically as a large-scale PDE. So you have an input that comes in, captured by the photoreceptors, and then the local computations and interactions result you to the output, a movie. And your retina, indeed, in the human retina, and most mammalian retinas, has around 20 or 30 such mosaics stacked in our eye, and therefore we have around 20 representations. And so what I will be talking about is this concept of cell type in relation to human diseases. And first, I'd like to convince you that most eye diseases are cell type specific for very simple reasons. And I will give you a very specific example today of a disease we recently discovered. It's recently discovered a mechanism, which is a disease of a computation within your eye. And then I'd like to talk about human disease models, how we now build up disease models for neurodevelopmental diseases of the eye. And finally, I will talk about our efforts to target cell types for repair in blinding and other conditions. So the key point is that these different cell types that drawn so beautifully by Cajal are not only different by morphology, but each has a unique gene expression pattern. And of course, it's very common knowledge that we have it all over in brain, but the retina is a special case, is that these gene expressions are really, really unique for these different morphologies. And therefore, very simply, if you have a mutation in a specific genes, and I like to mention that there are many monogenic diseases of the eye, so if you have a mutation of such that hits a cell type specific gene, and this cell type is important for the function of that circuitry, of that movie that it computes, then one might expect to get a disease symptom. Probably not explain all the symptoms as we heard, but a specific disease symptom. So all what we have to do is somehow learn what is the barcode for gene expression. And we and several other groups are embarked on it many years ago to have an atlas of gene expression for different cell types. We were very lucky because NIH had a large-scale program, the GeneSet project at Rockefeller University, and we get all the eyes from this program. So we reconstructed the retinas from around 1,500 retinas, and then pulled out around 100 mice with different cell types labeled, and then from those we took fact sorted out the cells and have an atlas of gene expression. And what we were really surprised is how specific are these expressions for different cell types. For each of them, we found around 20 or 30 genes that were specifically expressed within the retina only on those neurons. Now from here, how do we get to disease is rather simple. We look at a human database where we have the list of genes implicated in monogenic diseases with its symptoms, and we look at our mouse database and merge them. We create a mapping, and by arguing that if a gene and a cell type is conserved, then we can go from human symptoms and maybe get mechanistic insight in cell types and circuits in the relevant mouse models. Now, so here is our first mapping. This map is actually much, much bigger today, but this was how it was we produced 8 years ago. So these are monogenic, these are genes in monogenic diseases. These are cell types in the retina. And what was really exciting for us, that apart from the photoreceptors, this is a retinitis pigmentosa group, there were several inhibitory neurons hit by different monogenic diseases. So here is our first hit. I'd like to show you this gene FRMD7 that is very specifically expressed in a single type of inhibitory neuron, this number 7, that we confirmed by in situ hybridization both in human and in mouse retinas. And if you look at the cell what it hits is a very famous cell. It's called the starburst hemacrene cell. So this is one out of, let's say, 50 inhibitory neuronal type in your eye and the mouse eye, about 0.3% of the retinal cells. And then we started to ask, what kind of symptoms these patients might have. And they have basically two major symptoms. The first is that the eye is vibrating in the horizontal direction. But the second symptom was more important for us. Here is a recording from a patient by a postdoc in my lab showing that the horizontal optokinetic reflex is completely missing. So this reflex is that one you use when you look out in the window of your car and the tree is going by and your eye will follow the trees and then jump back and follow and jump back to stabilize the gaze and stabilize the image on the retina. So what your eye would do is that when you have horizontal motion, it follows the scene and jumps back. And this is completely missing in patients. What you see is these oscillations. Indeed, in the stimulus power, there's zero follow. However, and that was the key insight, that my postdoc asked the patients to actually follow voluntarily the stripes. They were beautiful and easily follow the stripes. So the reflex is missing, but the motor system and the voluntary motion is completely there. Amusingly, if you do the same experiment in the vertical domain, everything is completely normal. So this is only the specific lack of horizontal optokinetic reflex, what you see here. So why were we so excited? Because we knew the starburst hemacrene cells are involved in a specific computation in the retina, which is the computation of the direction of motion. So this is a discovery by Horace Barlow 50 years ago in Berkeley that he showed to a specific ganglion cell type in the retina, which is the output neurons of the retina. He's showing motion stimuli, and he noticed at that time in rabbits that there are some direction when there are a lot of spikes. He called preferred direction, and there is very few. He called it null direction. Now, his key insight was then he started to enumerate which directions are present in the retina, and each arrow now represents a directional selective cell. Now, this is recorded actually in our lab, but it's the same observation, is that you see 4 major directions and around 7 different ganglion cell types doing that. And in the last 50 years, the field that sort of emerged on sort of developed a very clear understanding what is how is it done, the computation of direction. And this depends on this starburst hemacrene cell, where actually the gene expressed, which is mutated. And these starburst cells, what they do, they dedicate the different processes. This is GABAergic cell. This has axons and dendrites in the same processes. And they dedicate different compartments of the processes to make synapses for the 4 different ganglion cell types. These are all genetically different. And because of this asymmetric hookup, what they have, that is how directional selectivity developed. Now, because, and we also knew that if you ablate starburst hemoclin cells, you can completely get rid of the optokinetic reflex. This circuitry is driving the optokinetic reflex. That's where we were excited. So we built a mouse model, and the mouse model really beautifully shows the complete lack of optokinetic reflex. However, interestingly, you see that only one of the symptoms is present in mouse. The vibration of the eye is not present, and I can, if somebody asks me why, we can talk about it later. Now, so we have one of the symptoms present, so we can ask now, what is happening in the retina, and then we can take a multi-electrode array, put down a normal mouse retina, and have you can see the 4 different directions, and then you put down the retina of the FRMD7 mutant, and you see an extremely clear diagnosis of the lack of this of horizontal optokinetic reflex, because the direction selective computation is entirely missing in the horizontal direction, why the vertical is completely normal, showing why the vertical is present and the horizontal optokinetic nystagmus is not present. Now, I don't want to spend a lot of time, but what we discovered, and this is already published, is that the reason you don't have direction selectivity is not a cell death or anything, that all the circuit components are present, but during development, the starburst hemoclin cell doesn't learn what is left and right, and is unable to make these asymmetric synapses with these 2 horizontal type of direction selective retinal ganglion cells. So here is a disease where we start from a patient and a gene, and we start to zoom in to a relevant symptom, and then we find a relevant cell type and the circuit, and this is a neurodevelopmental disease because all this developed before eye opening. Now the question in such a disease is that if we know the circuitry, how do we get potentially towards curing it, towards understanding the molecular mechanism and coming back to patients? And one of the clear problems is that, of course, what we want to do now is looking at the molecular mechanism, how FRMD7 is involved in this decision of left to right connectivity. However, and it's quite easy to do because you pull out the binding partners and others in mouse. However, we really want to know if the genes that we pull out in the pathway are present in human. So that puts us in a position that we want to understand a unique cell type in the retina during development in a human. So how do we get access? And this is what I like to talk about now, is that new developments to develop human models, cell type specific models, where we can really access the same cell types during development in a human scenario. So this all depends on major discoveries by Yoshiki Sasai, who observed that if you take fibroblast skin biopsies from skin biopsies and turn them into iPS, and you grow them in the right condition, the eye field, the eye cups are growing up from these human cell mass. So that was the basic discovery and the most important one. And so what the field and us and some other people started to do is cutting off these optic cups from these human cell masses and started to grow them. And when you grow them, what you notice is that each of them is developing to a sheet, what we call is a retinal organoid. And I will show you soon what happens sort of month by month in these organoids. But the key point I like to make is that you can grow a hell of a lot of these organoids. From a single human, we can grow, let's say, 1,000 retinas or 1,000,000 retinas. This is simply a matter of scaling. What matters a lot is the initial clone, iPS clone. If you get a good clone, you can grow any number of retinas. Now, so what happens, let's say, at day 42, now we cut a section from the retina, and you can beautifully see that these retinas start to stratify. You can see the photoreceptor, developing photoreceptors on one end, developing ganglions on the end, and in between, there are some other cells. Okay, so this is very, beginning of the story, but at day 125, we notice that completely beautiful human cone photoreceptors develop. Here is the axon, the terminal, the inner segment, and the starting outer segment. They have beautiful cilia, so we can actually model all cilia diseases in this case. They have also beautiful rebound synapses right there. And I don't show you that we have 3D EM data showing you that beautifully they form synapses with bipolar cells, just as they form in any normal, real human retina. Now, of course, this is only photoreceptors, and what we want to access is these 80 cell types, what I showed you. So we keep growing them. And so we have to modify protocols. But what happens in day 210 is that a beautiful, really three-layered human retina, photoreceptors by its inner nuclear layer, the ganglion cell layer, or you see the two plexiform layers develop. So we can now ask the question that at these late stages, what sorts of cell types do we have? And amusingly, one by one, we can find essentially all cell types that we normally find in a well-respected normal retina. For example, here are the starburst cells, which is 0.3% of all your retinal cells, are beautifully labeled in these human retinas. And not only one marker works, but essentially many other markers. Here are some others showing that these are developing cell type, very similar what we have humans. And now what we're doing is, of course, doing knock-ins, knockouts, and labeling cell types to purify these cells and looking at the mechanistically how the disease develops. Now, the only problem with this was that the ganglion cell side, when they grow, they have normally axons growing into the brain. But here, we just have alone a retina, and the ganglion cells, over time, they die, they degenerate, because their axons had nowhere to go. And this is where the development of eye-brain organoids from Vienna helped a lot, that we can now develop these eyes hooked up with the brain organoids by simply growing them longer. And by now, we have really beautiful ganglion cells with all the axons labeled growing into the brain. So we can have access both to the inhibitor neurons as well as the ganglion cells here. So we can model a large number of human diseases, including glaucomas and optic atrophies. Many of them we are sort of growing up in the lab right now. So in the first part, I talked about diseases that hitting cell types and how our attempts to build human models. But at the end of the day, we're interested in therapy. And one way for therapy is targeting cell types in different diseases. Not every disease would it work. We are especially interested to regain vision in certain kind of blindnesses. And for what we need is vectors, AAV vectors we can inject to the eye, which are completely specifically expressed in different cell types. And so what we want to do is really reverse the process, now go from mechanistic insight in cell types and circuits to move to remove symptoms in human genetic diseases. And for us, the key problem was really how to target cell types by AAVs. And what we went back is the same idea as at the beginning I showed you, that these cell types have very unique gene expression pattern. And we argue that if they have unique gene expression patterns, then we must be able to find enhancer element that smell out this gene expression, the local gene expression, and report it. So we started to build large-scale AAV viruses with different enhancer based in our gene expression atlas and ataxic data and other kind of data that I don't want to get into. All what I say, that there is a mapping that using a heuristic, we create enhancers, then created 260 different viruses and all tested in vivo starting in mouse. And now we are doing it in monkeys and humans. And turns out that we don't fully understand why and which enhancers work, but around 1 out of 10 enhancers incredibly precisely is labeling cell types. And I just want to show you some examples, is that this is a single injection in a mouse eye. We label for the rest of the life of the mouse only and specifically cones, none of the rods and none of the other 70 cell types. Here we can label only the rods and none of the cones. Or then we can label rods and cones and absolutely nothing else. Or we can label a single type of inhibitory neuron, or ganglion cells completely specifically, or Muller cells, and I can go on and on. At least 20 cases we have, we completely specifically label only a single cell type, so we don't have to do any more genetic knockouts or genetic knock-ins with Cree and others. We can just use these vectors to bring in anything that fits an AV. Now, this is only a mouse, and of course, the big question is that how to do it in a human. And again, technology helped us, and the technology came from keeping human retinas post-mortem alive. So this is originally coming from Serge Picot, my colleague in Paris, who observed that from human donors, he could keep human retinas alive for around 3 weeks. And meanwhile, technology got better, so by now we can keep human retinas alive for 10 to 12 weeks, and now we can test these vectors, indeed all of them. And here just showing the first hit, we can completely specifically target rods, here cones, and we have many, many other reporters we can target. So I'd just like to finish that we are using these targeting methods now in a disease called retinitis pigmentosa, it's clouding blindness, and we try to bring in optogenetic sensor to these cells to recover vision, and we have a clinical trial soon starting with GeneSight Biologics in collaboration with Jose Sahel, Serge Picot, and Denis Dalcara. So we are moving from mouse all the way to patients with the first clinical trial set to the end of this year. So I'd like to finish going back to my original, or the original drawing of Cajal, that I showed you an example of a human disease that is localized to a particular inhibitory neuron. Turns out that most of these inhibitory neurons are there to detect motion. And if you go to an ophthalmologist, you notice that the ophthalmologist will not test you any motion diseases. So I argue that there are a lot of diseases out there that are hitting these different inhibitory neurons. We just not yet diagnose them. So what we need is not only more human genetics to find more genes, but we need completely new tests in ophthalmology to have the ability to find motion diseases and then figure out that how it's localized to circuits. Thank you very much.
How functional are these cells, okay, in the retinal organoid? And so you also show that retinal brain organoid. So how is the innervation patterns? You know, there's a presumptive lateral geniculate body is now target.
So the first question is, in the mouse retina, we recently published that we were able to achieve light sensitivity in the photoreceptors. So we identified the molecular pathway, which is growing outer segments. And we're shown in mouse that they have light responses, the photoreceptors. Now in human retina, we are in the process to do that. So I think this is not so hard to do. One just have to tweak it to get it. Now, we are currently recording from these and looking at the synapses, you know, it's around the age of 200 days in humans. Several synapses are already made, some of them are not yet, so it's just a question of time. Now, regarding to projection to brain, looking at LGN, the presence, it's an ongoing work. We just have a line where we have a ganglion cell specifically labeled, and we have channel rhodopsin, so we are looking at which areas, and we engineered in GChem, so we are looking at which areas activated and whether we can find any of the known targets. This is not yet clear.
Oh, interesting. Okay, thank you.
Reflect the mutations that you see in humans, and are they preserved, and do they show something functionally?
Right, so this is what we are looking at. We just succeeded to build these retinas completely. So we have around 20 genetic diseases we engineered in, and we are making around another 50 and looking for phenotypes. So this is not yet done. For many, from FMD7 and many others, we already have the organoids, and we are carefully looking is that what is going wrong. Certainly at the molecular level, we can detect the problems, and now we are looking, for example, whether we get rod cell death or cone cell death in Leber congenital amaurosis or Usher and other syndromes we are looking at. I think we are at the point where we have all these cell types, And the next wave is to see which mutations will represent a disease pathology. But even just for us to have the ability to separate these cell types and confirm that actually the genetic networks are actually very close to the normal human, because we have access to normal human retinas and one can see is already very reassuring that we are looking at a sort of a state which is not so far from the human state.